everyone, this is Heather Benson. I'm from Team Go Genetics, but I'm flying solo this week because my group member had to drop the class. So it'll just be me. This is lesson 3, applying genetics problems, and I'm going to do a screen share so that you guys can follow along with what I did to solve these problems. Okay, so for problem number 1, it talks about epigenetic inheritance. And for the first question, it talks about Angelman syndrome and how that works with genomic imprinting. In the case of Angelman syndrome, the imprint silences the paternally inherited alleles. Suppose lowercase a is the Angelman syndrome allele, while uppercase A is the non-disease allele. The following cross is done: heterozygous male times heterozygous female. What are the genotypes and phenotypes of the progeny? So for this one, we can see that we have the female, and here we have the male. And when they cross, it turns out the genotypes become 1/4 homozygous dominant, half heterozygous, and 1/4 homozygous recessive. And the phenotypes, you can see here which genes are silenced, and that's because those genes come from the paternally inherited alleles. So half of the progeny will have Angelman syndrome and half will be non-diseased. For part B, what are the genotypes and phenotypes of the progeny of this cross here? So we have homozygous recessive and heterozygous, and the heterozygous is the male. And so because this capital A and this capital A and this lowercase a and this lowercase a are silenced, it'll be 100% Angelman syndrome because the lowercase a, as indicated up here, is Angelman syndrome. For number C, for letter C there, it is talking about what happens when a woman who has triplo X syndrome, how many bar bodies does she have if the XICs have been deleted from 2 of her chromosomes in each of her cells. And so for this one here, 2 XICs must be present in order for 1 X chromosome to be inactivated. So since there's only 1, there would be 3 bar bodies present in each cell. For part D, suppose that you isolate a cell from a female cat with 2 X chromosomes. However, the T-SIX on each X chromosome are non-functional, while the XIST genes on each X chromosome are functional. How many bar bodies are in the cell? Well, this would be 1 bar body in the cell because the lack of function from the 2 T6s does not affect the formation of bar bodies. The XISTs both work. They still coat and inactivate 1 of the X chromosomes. Okay, so for question number 2, SRY is an example of a holoandric gene. MIC2 is an example of a gene found on both the X and Y chromosomes. And in this cross between the IGF2 silenced male mouse and an IGF2 silenced female mouse, the phenotypes of the offspring, as shown by this cross here, half will be normal, half will be dwarf. And that's shown here. And then in part D, in the sex influence pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other sex. Genomic imprinting is an epigenetic process that occurs during gamete formation, while dosage compensation is an epigenetic process that occurs during embryogenesis. Letter F, one of the topics discussed in lesson 2 involved the formation of a double-stranded RNA molecule. What 2 molecules form the 2 halves of this double-stranded RNA? And we learned that that's XIST and T6. When reciprocal crosses give identical results, the alleles that control the trait in the question are located on the autosomal gene. When reciprocal crosses give different results, the alleles that control the trait in question are located on the X chromosome. So that's it for question 2. For question number 3, this is talking about the OTD syndrome. And so this is the woman's mother is homozygous and unaffected, and the father has the disease. So that's shown right here. The father has the disease, and it's a recessive disorder, so lowercase. Mother's not affected. So the child, this is the daughter's genotype, she's not affected. However, in part B here, if she has children with a man who does not have it, shown here, what's the chance that her first child with a son will be born with OTD? And that would be, well, that would be a 25% chance. She, out of the 2 sons, only 1, so 50% of sons would have the disease, so only 25% of all children. For part C up here, what genotype of a male with whom the woman could produce a daughter with OTD? And that would be, they would have to have the same genotype, the same genotype on the X there. And then that would cause this daughter here to have homozygous recessive on both of those X's. And so she would have the disease. In part D, consider the crosses in part C. What are the expected genotypes and phenotypes? This here would be a daughter with OTD. This here would be an unaffected daughter because it would have to be on both in order for her to be affected. The son, he has the disease, and this son here does not have the disease. Okay, on question 4, this is talking about the presence of a beard in some species of goats. It's determined by an autosomal gene that is dominant in males and recessive in females. So suppose a cross produces a 3-beard, 1 no-beard phenotype ratio among the male offspring. What are the genotypes and phenotypes of the parents? So in order for that to happen, the genotypes would both have to be heterozygous, and the mother would have to have no beard and the father would have to have a beard. And that would produce, all 3 of these here are beards. And this here is no beard. In part B, this would be the same genotype for the parents, because this here is the only genotype possible for a female goat to have a beard. In part C, suppose a cross between a male with a beard and a female without a beard produce a 1 beard, 1 no beard phenotype ratio among the female offspring. What are the genotypes of the parents? Well, as you can see here, this is the only no beard. This is the only beard that can come from that. What are the genotypes of the parents? Homozygous, recessive, and heterozygous. The mother would have no beard and the father would have a beard. Okay, question 4D. Suppose a male and a female, both which lack beards, produce a 1 beard, 1 no beard ratio among the male offspring. What are the genotypes of their parents? Well, we have 1 beard, 1 no beard. So the mother would be heterozygous, no beard, and the father would be homozygous recessive, no beard. Okay, so on question 5, this is the one about cats, and in cats, orange coat color is determined by the B allele, and black coat color is determined by the uppercase B allele. The heterozygous condition produces a phenotype known as tortoiseshell. The uppercase B and lowercase b alleles are X-linked. Suppose you cross an orange male cat to a tortoiseshell female cat. What are the genotypes and phenotypes of the progeny? So we have that here. We have the indicated female and the indicated male. We cross here. This would be female tortoiseshell. This would be orange female. This would be black male, and this would be orange male. On B, they want to know, male tortoiseshell cats are very rare. What is the genotype of a male tortoiseshell cat? Well, tortoiseshells have to have heterozygous alleles in order to exist. So in order for a male cat to be a tortoiseshell, it would have to have an extra X chromosome, which is known as Klinefelter syndrome. So that's shown here, the only possible genotype for that to occur in a male cat. Part C, in the fruit fly Drosophila, a mutation produces the scalloped phenotype, irregular margins on the wings. Assume that the scalloped phenotype is the result of an X-linked recessive mutation. Indicate the genotypes and phenotypes of the progeny produced when the reciprocal crosses are done. So this here shows the normal cross indicated. And when you do the reciprocal cross, the genotypes and phenotypes would be 50% homozygous dominant, female no mutation, and 50% here with the capital R, male no mutation. Question 5D up here. This question concerns scalloped phenotype described in Part C. Assume that the scalloped phenotype is the result of an autosomal recessive mutation. Indicate the phenotypes and genotypes of the progeny produced when reciprocal crosses are done. So here is the normal cross, and it shows 100% there, 100% of the mutation. So when you do the reciprocal cross, the genotype would be 100% homozygous dominant, and the phenotype would be 100% no mutation in both males and females, which was unlike this cross over here. Okay, well that's it, guys. So let me just turn off my screen share here. Thanks, guys. See you later.